Hi, my name is Carissa and this is my BRCA gene final project. So in our lab we learned about breast cancer and I'm going to be focusing on how it connects to 4 units of AP biology. Firstly, BRCA1 and BRCA2 are tumor suppressor genes that I will talk more about later on. Being able to understand the role of these genes is crucial for genetic screening and cancer prevention, as many factors play a role in how breast cancer is diagnosed in a patient. The objective of the lab was to identify BRCA gene mutations in DNA samples from our patients. This was done using gel electrophoresis, and from the gel, we were able to determine which patient carried mutations associated with an increased risk for breast cancer. This case focuses on the Altermans Hannah, her older sisters Sarah and Rachel, and her mother Deborah. Rachel was recently diagnosed with breast cancer at the age of 30. Her mom was diagnosed when she was 42, only 5 years after Hannah had been born. Her other sister Sarah is 25 and does not currently have breast cancer. Because of their family history of breast cancer, they all met with a genetic counselor in order to test for BRCA mutations in Hannah and Sarah. So the first unit I chose is Unit 3, and Unit 3 covers cellular energetics and includes the topics of enzyme structure and catalysis, environmental impacts on enzyme function, pH competitive and non-competitive inhibition, cellular energy, which is ATP, photosynthesis, and cellular respiration. This unit connects with our focus of the lab because it highlights the processes of DNA replication and repair and also involves the use of enzymes and their role in cellular energetics. Once the DNA samples of each patient are placed in the wells of the gel, restriction enzymes work by scanning along the length of DNA, looking for a particular sequence of bases they recognize. Once it's located, the enzyme will attach to the DNA molecule and cut each strand of the double helix. Eventually, the full length of the DNA molecule will become broken into fragments. When the DNA is placed into the gel, they are separated by the electrophoresis and the fragments migrate across the agarose gel. With the help of restriction enzymes, we were able to identify any mutations based on the resulting patterns. The next unit I chose is Unit 4. And Unit 4 is about cell cycle and cell communication. So Unit 4 includes cell communication, signal transduction, and ligands and receptors, feedback, including positive and negative feedback loops. Positive feedback increases the effect on a stimulus, such as child labor, and negative feedback reduces the effect of a stimulus. So in response to being warm, you'll sweat to cool down. And it also covers the cell cycle and how it's regulated. The cell cycle is regulated by checkpoints that help the cell decide if it will progress to the next stage of the cell cycle. These checkpoints can be controlled by signals inside the cell, such as cyclins and cyclin-dependent kinases, or outside the cell by growth factors. Signal transduction pathways help move the cell in and out of different stages of the cell cycle. When the cell signaling is not functioning properly, this is when cancer can occur. So how does this unit connect with the lab? Because there's a family history of breast cancer in the Altermans, genomic biomarkers were used to help with their genetic testing. So BRCA1 and BRCA2 are tumor suppressor genes that help to repair damaged DNA and help prevent cancer when they are working normally. However, the problem occurs when there are mutations in these genes and or other genes that have been identified to be linked to breast cancer. Due to the Altermans family history, they are candidates for the BRCA2 biomarker testing, and I'll go more into depth later as to why and the history of this. So in the lab, I focused on finding mutations for the BRCA2 gene. Cancer is a disease of uncontrolled cell division, and its development and progression is usually linked to a series of changes in the activity of cell cycle regulators. In Unit 4, we learned that inhibitors of the cell cycle keep cells from dividing when conditions aren't right. So too little activity of these inhibitors can promote cancer. Similarly, positive regulators of cell division can lead to cancer if they are too active. In normal cells, the BRCA genes help make proteins to repair damaged DNA, but mutated versions of these genes can lead to abnormal cell growth, which can lead to cancer. Mutations in the BRCA genes affect cell cycle regulation, and it really highlights the role of both BRCA proteins in the DNA repair process and cell cycle regulation because it's critical for maintaining cellular function and just preventing cancer in our cells. The next unit I chose was Unit 5, and Unit 5 focuses on heredity. And the topics it includes are meiosis and genetic diversity, Mendelian genetics, alleles, genes, Punnett squares, pedigrees, and non-Mendelian genetics, such as X-linked inheritance. It also covers environmental effects on phenotype and chromosomal inheritance. This connects with Unit 5 because breast cancer can be a genetically inherited mutation. And based off of the pedigree I made, BRCA is not confined to a single generation. It is also observed in both male and female members of the Alterman family. And this indicates that the gene is not sex-linked to females, so it must be autosomal. And because the disease is transmitted from both affected and unaffected parents to their children, we can infer that BRCA's inheritance pattern is also due to a dominant allele, which means one copy of the altered gene in each is sufficient to increase a person's chance of developing cancer. And as I previously established before in my Unit 4 slide, the Altermans are a candidate for BRCA2 biomarker testing because BRCA was found in both males and females based off of their family's pedigree. Mutations in BRCA2 biomarker testing are more likely when both females and males are affected, whereas BRCA1 mutations tend to be used with families of incidence of both breast and ovarian cancer. Unit 5 connects with the main focus of this lab because we're using pedigrees and alleles to understand the genetic history of the Altermans and conclude the results of the gel electrophoresis. After the gel finished running, I compared each gel lane to the marker. If the fragment lengths corresponded to a certain allele that's associated with the BRCA2 mutation, then I would know the individual carries the mutation. Based on the gel electrophoresis results, Hannah has one of the BRCA2 alleles that Rachel and Deborah have, and they also had similar RF values and fragment lengths. And although she doesn't currently show signs of having breast cancer, this could indicate that she has an increased risk of developing breast cancer. And on the other hand, Sarah's gel electrophoresis results were different from Rachel and Deborah's because she has two normal alleles, so this likely means that she has a lower genetic risk associated with developing breast cancer. And if you look at my Punnett squares, Deborah had the dominant allele for breast cancer, while Hannah and Sarah's father had two recessive alleles. So based off of their cross results, 50% of their offspring would have the dominant allele for breast cancer, and 50% would have the homozygous recessive allele. So it's likely that Rachel and Hannah could have the dominant allele for breast cancer, and Sarah has the homozygous recessive. So the next unit I chose was Unit 7, and Unit 7 covers natural selection and evolution. The sections and topics it includes are natural and artificial selection, population genetics, Hardy-Weinberg equilibrium, and Hardy-Weinberg equilibrium is the constant frequency of alleles in a gene pool, evidence for evolution, common ancestry such as homologous, analogous, and vestigial structures, phylogeny, which is the representation of evolutionary relationships among various species, and speciation, which is the formation of new species. It also covers extinction, variations in populations, such as genetic differences among organisms, and just the origins of life on Earth. So back to the lab. Founder effect is a type of genetic drift that occurs when a small group leaves a larger population and starts a new colony. When groups of individuals leave initially large populations, certain genes tend to increase in those smaller populations, and then individuals pass those genes or mutations of those genes to others in the population. So because of a founder effect / mutation that occurred among Okazaki Jewish, this event caused a high prevalence of BRCA1 and BRCA2 inherited gene mutations in families of Okazaki Jewish descent. So in the lab, the BRCA2 gene is present in the Alterman family as they are of Okazaki Jewish descent. And this could explain why breast cancer runs through their family. So my last slide is my bioethics slide. And one bioethical concern associated with breast cancer is the disparities in access for breast cancer care and treatment. So many cancer patients are unable to receive care for a number of reasons, whether it's due to race, poverty, or geographic disparities. They all play a role in preventing many individuals from receiving proper care they need. So I took two perspectives, one from a doctor and one from a patient with breast cancer. So I found a story online of a surgical resident specializing in oncological surgery, working in a public hospital located in Mexico. She found herself in an ethical dilemma about what treatment options to offer patients with breast cancer. And when discussing treatment options, she wondered, should patients be offered hard to access or prohibitively expensive options? She explains to her patients that they can perform a conservative surgery, but radiotherapy is not available at their public hospital. Therefore, patients must be sent to a hospital in another state about an hour from their homes. So due to the high volumes of patients being sent to this hospital for radiotherapy, the wait times are long and many patients end up not receiving the treatment they need within the recommended time frame. So this doctor is faced with another dilemma. Propose to patients to the two therapeutic options, which I'll explain in a second, or omit the surgery plus radiotherapy in another hospital. Since it's difficult to access and costly, the doctor knows it's highly likely that patients will not get radiotherapy in time and will be more likely to suffer a reoccurrence of cancer. By offering only the second option, the doctor would prevent them from considering the surgery option, but ensure a good chance of recovery. So because of the situation, the doctor wonders if a doctor should always tell the whole truth, even if this has the consequence of offering an option, if chosen by the patient, because it could be highly likely to result in a worse prognosis due to the lack of timely access of receiving this treatment. So my second perspective from the point of view of a patient that's facing this bioethical concern is this patient has been diagnosed with early stages of a cancer progression, and they have two therapeutic options. One, surgery that conserves some breast tissue plus radiation therapy, and radical surgery. So while both options offer a roughly equal chance of healing, after surgery, the patient would have a maximum period of 8 weeks to receive their radiotherapy. And if the radiotherapy is not completed, because they have to wait a long time before they can go to this other hospital or it's too expensive, then the chance of cancer reoccurring is high. So overall, the availability of resources limits doctors' and patients' options. Poor or low-income patients are more vulnerable and more impacted by a situation like this due to their socioeconomic condition and their physical condition as cancer patients. So that's all I have. Thank you.